I know there's probably a pretty diverse set of backgrounds here, so I'm going to start with a pretty broad introduction to next-generation sequencing, assuming you know nothing about it. And I'll go on to discuss some potential applications in lab medicine. And I'm going to come back to some work that is ongoing in my own lab and other labs in genome sciences with some potential relevance. And I'll then just try to summarize and wrap up. So we're just over 3 decades past the invention by Fred Sanger and independently by Wally Gilbert of what's called electrophoretic sequencing. So this is the standard method of sequencing for 3 decades and what was essentially used for the Human Genome Project. And the basic idea, as I'm sure many of you know, is that you have chain termination with dideoxy nucleotides. In the old days, you'd run 4 lanes corresponding to A, G, C, and T of the products of these reactions. In the mid-80s to 90s, this was automated, and also a fluorescence-based detection system was developed, resulting in traces that we're much more familiar with. And overall, through the automation of this process and its essential scale-up and optimization through the Human Genome Project, there was essentially an exponential increase in the throughput of sequencing over several decades. And this is shown here by a graph of the overall amount of sequence deposited in GenBank. And starting from essentially the first paper on sequencing, which included a report on the genome of a bacteriophage, the whole project, or the community as a whole, worked its way up the food chain, culminating in the Human Genome Project and subsequently moving on to other genomes of equivalent size using the infrastructure that had been developed over these years. So one can, or the analogy has often been made between sequencing and Moore's Law, which is primarily was conceived of in relation to semiconductors. But when you plot the throughput of sequencing and the cost of sequencing, they essentially follow exponential trends similar to that. As you can see here by this number of websites trend, exponential trends are not guaranteed to last forever. And one of the things that scientists noticed, or the genomics community noted sort of towards in the years following the Human Genome Project, was that nearly everything that could be optimized was optimized. And people were essentially running out of ideas in terms of how to continue the trends that had been maintained for 3 decades. And so that spurred a number of groups, including the lab where I was in graduate school, George Church's lab, to think about alternative approaches to DNA sequencing. So some of the ideas that have actually been bandied about since the 80s, or since the early days of sequencing, include 4 that I'm showing up here. So one which is nanopore sequencing. The basic idea here is to drag DNA molecules through biological or synthetic pores and look at perturbations in current as a way of figuring out what the sequence is. Another idea was simply to multiplex the living heck out of Sanger sequencing. So here we have 384 essentially capillary gels, all pointing towards the center, and this can essentially is like a 384-plex Sanger sequencing reaction. People pursued microarray-based sequencing as another approach. And then the approach I'm going to focus on today can be lumped under a single term, cyclic array sequencing. And so I'll tell you a little more detail what I mean by this. So the basic idea here, we tried the term cyclic array sequencing pretty early on, but it didn't really stick. And so people tend to refer to this as either next-generation sequencing, second-generation sequencing, or massively parallel DNA sequencing. And the basic concept here is that you have an array, so this could be a glass slide or something like that, of tethered features. So imagine that each of these circles corresponds to one fragment of DNA, several thousand copies of one fragment of DNA, and the copies are locally clustered and physically tethered to the surface of the array. Right? Now, through some biochemical method, so for example, polymerase extension with 4 fluorescent nucleotides, one can interrogate the identity of a position in each feature of the array. So for example, if each of these features on the array consisted of prime templates, and we added a polymerase and 4 fluorescent nucleotides, we would expect that each feature would light up one of 4 colors. And then we could go through and snap a bunch of pictures of the array, and we'd essentially have sequenced a single base. But then we could come back and repeat it again for the second base, go through again on the array and take a bunch of pictures, the third base, and so on and so forth. And essentially, by observing this array over the course of several cycles and looking at the behavior of each feature, one can construct continuous sequences that correspond to the sequence of that particular feature. So in practice, so here on this kind of mock example, I'm only showing a handful of features on the array. But in practice, one can potentially fit millions to billions of independent DNA sequencing features on a relatively small array. So here, I'm showing about 1/10,000th of the area of a microscope slide. And you can theoretically pack as many as 2 billion beads or sequencing features onto the surface of a standard microscope slide. So the reason this is so much cheaper than conventional or Sanger sequencing is one, because it's remarkably multiplex. So here we're going to be sequencing these millions or hundreds of millions of sequencing features in parallel. And the second reason is that when we, for example, wash a polymerase and 4 fluorescent nucleotides over the surface of the array, we're using a microliter scale volume, right? But because we're adding nucleotides to all of these millions of features in parallel, the effective volume per sequencing feature drops to the level of picoliters or femtoliters. So the cost saving is essentially realized there. So this is actually an image from my PhD work. These are 1 micron beads that are lit up one of 4 colors. And again, this is approximately 1/10,000th of the area of a microscope slide. So what these technologies have essentially allowed us to do is to continue the trend that has been in place for 3 decades. So early on in the mid-90s, remember these gels and doing sequencing by hand. A single person could process hundreds of nucleotides of sequence a day. With the early implementations of automation of these technologies, went up to thousands. The Human Genome Project motivated extensive optimization of the automation such that instruments were producing millions of bases today. And today, any one of the 15 to 20 next-gen sequencers that are on the UW campus can each produce billions of nucleotides per day of sequence. So the commercialization of these approaches, of these new technologies, has proceeded pretty rapidly. So here are a bunch of companies these second-gen sequencing companies that are all pursuing some variant of the basic schematic of massively parallel sequencing that I showed on an earlier slide. They all differ in the details. So how they're generating those sequencing features and how they're actually doing the sequencing differs. So some of these companies are using polymerases as a sequencer. Some are using ligases. Some are sequencing single molecules. But they're all basically employing the same approach of trying to sequence millions of independent features on a 2-dimensional array in parallel. There are also a few companies that are pursuing approaches that are far more out of the box or out of left field than the ones up here, and these include Pacific Biosciences and Visagen. So I'd put those in a category of so-called third-generation sequencing, but they're not yet commercially available instruments. So there is a cost. So on one hand, we're getting hundreds of millions of sequencing reads in parallel now, and it's 1,000-fold cheaper than Sanger sequencing, let's say, approximately. The costs are evolving very quickly. But the downsides are that, by and large, the reads are much shorter. So a Sanger sequencing read is on the order of 1,000 nucleotides. Next-gen sequencing read can range from 30 to 100 nucleotides, depending on the platform. And the accuracy is also considerably worse. So those are downsides, right? So it comes at a cost. It also suffers from what you can call poor granularity. So if one wanted to do a Sanger sequencing reaction, for example, one would sequence 1,000 bases and get 1,000 bases of data, right? The minimum unit of sequencing that you get with next-gen sequencing is on the order of let's say, 1 billion nucleotides of data. And there's no easy way to get just 1,000 bases or just 100,000 bases. And so as a consequence, that impacts the way in which applications are actually put into practice. And then the turnaround time is also can be long, right? So running these instruments can take days to weeks. So before you get your data back, right? Which has implications potentially for clinical applications. So here's just a screenshot of some data. So this is human genome resequencing. So down here at the bottom, we have the reference human genome. And so the nice part about applying these new technologies is that even though the reads are short, it doesn't matter much in the context of trying to resequence individual humans, because we already have a reference genome from which we deviate only slightly as individuals. So mapping these reads to this reference genome is a remarkably simpler task than assembling this genome in the first place. And then the bioinformatic challenge becomes distinguishing the many errors from actual positions where there is a variant, right? And then obviously turning these variants into functional predictions about what they would, for example, do to a protein sequence. So, so how can this potentially be applied? So if you think about the myriad of ways that it's been applied in research already, I think we're moving towards kind of a world in which DNA is increasingly thought of as a single format or a machine-readable format for capturing biological information at high throughput. So not just sequence of genomes, but also transcriptomes, so looking at RNA expression, looking at protein-DNA interactions, looking at chromosome conformation, looking at methylation, and the list goes on in terms of the various kinds of information, biological information, that people have found ways to translate into sequence data that can then be captured on the same instrument, right? So I think, in thinking about how this might be applied clinically, and I think that it's not being applied clinically very much at this point, I think you kind of have the same motif, which is that there are a range of applications in which there might be relevance of these technologies in the short run or long run for clinical use. So the one I'm going to start with is because this is the one that's been bandied about the most in terms of what's been talked about, is personal genomics. So the basic idea here is that, branching off the Human Genome Project and the model organisms that had their reference genome sequence, that we can now sequence genomes of individuals for which a reference is available. And the cost of sequencing individual genomes has dropped considerably just in the 8 or 9 years since the Human Genome Project was completed. And even this slide is a bit outdated. Today you can, if you're sufficiently motivated and have the spare cash, you could buy your genome for $50,000, all right? There's a number of companies popping up that are offering direct-to-consumer genomics. So most prominently in the news is 23andMe, but a number of other companies as well. And these are the subject of litigation in California and that sort of thing. But it's an interesting world we're stepping into. And so just to kind of put a thought slide up here. So personal genomes for the insured masses. So people are talking about this as, if the cost was to come down to $1,000 per individual, would it be worthwhile to make this kind of a standard part of healthcare? In other words, to sequence everyone. So today already we can capture the vast majority of variation present in any individual genome for less than $1,000. So just using a standard genotyping array, which costs less than $500 now, you can capture that information. But in the future, I'm sure you can do whole genomes for a roughly equivalent cost. So if you figure that we spend about $5,000 a year per person on healthcare in this country, and a $1,000 genome, and you only need to sequence it once, and you amortize it, then it boils down to the cost, and less than the cost of a magazine subscription, right? So only $13 a year. Of course, it's worth it, right? But the catch here, so there have been a number of individuals now who've had their whole genome sequenced. So for example, the first one I believe that was sequenced using next-gen technology was Jim Watson, and I'll bet you Jim might actually agree, is that the amount of clinical information that's actually gained based on what we know today may not even be worth that $13 per year, right? So we know so little right now for a relatively healthy individual. We know very little about the genome, such that full knowledge of genetic variation would impact an individual's the way their healthcare is managed in a clinically meaningful way. So this relates to an issue that sort of the field as a whole is struggling with, which is why are we interested in human genetics in the first place? And this is actually an active debate. So one view of the field is that gene-disease associations are clinically useful. So this is thinking about common variants that are associated with common diseases because they inform risk profiles in clinical decision making, or they might. And the other view of the field is that this was not the point. We're not looking for variants that influence human disease in order to develop diagnostics. Rather, we're trying to learn about biology. And so this essentially, the reason we're having this debate is largely because of the outcome of genome-wide association studies, which have been going on for the last several years, and were based on the premise that common variants have a significant impact on the heritability of common diseases. And it's kind of a glass half empty, glass half full situation where there are lots and lots of very real associations, but the variants that you see, or the variants that you find that are convincingly associated with a given trait, only confer a very small magnitude of relative risk. So in the same article at the same moment, you have two experts in the field, one saying this is, we're doing great, and the other saying that this is horrible, right? So, and they're both right, depending on how you look at this glass. So the kind of, just to wrap up the personal genomics point, I think that this area is getting a lot of hype, but it's still a long road before we're there, or before this makes sense to do on a routine basis for lots of people. And I think that rather we should be thinking about lots of other areas in which these technologies might be applied that make a lot more sense in the short term and might be more justified and more clinically relevant. So I'm going to talk about briefly some of those. So one is cancer genomics. So here's, in contrast with much of what has been learned from studying germline variation, mutation status, there are some clear examples where it's clinically relevant. So for example, this is a paper from a couple weeks ago in the New England Journal showing that therapy for basically whether you're using carboplatin, paclitaxel, or gefitinib is or should be informed by your EGFR mutation status. So EGFR mutation negative patients basically respond better to one therapy, and EGFR mutation positive patients respond better to the other therapy. So there's a clear rationale for sequencing here as influencing therapeutic choices. So you could just do sequencing on this, right? Sanger sequencing, PCR it up and sequence it. So why do you need next-gen sequencing? So one example of where this could be relevant is a paper from Matt Meyerson's group at Dana-Farber from a few years ago, showing basically that, so think about cancer. Cancer is heterogeneous. You're going to have stromal tissue contaminating your samples, and you also have heterogeneity within the tumor itself. So here is detection of a EGFR kinase mutation, but you'll note here is that we're looking at a mutation that is only present in a very small fraction of the reads, and in order to really see this, you need to essentially obtain tens of thousands of sequencing reads that independently cover the same position to be able to confidently distinguish it from the noise, right? And so I think, to think about this a little further beyond just thinking about one gene, personal genomics may be much more relevant to cancer. You can imagine a scenario in which we sequence whole genomes, or just the exons of tumor and normal tissue in challenging patients with cancer, identify point mutations and structural variations at all scales. So you're essentially doing CGH at the same time that you're obtaining nucleotide level resolution on mutations, and then intersect this with what we know about cancer pathways and identify treatments that are based on mechanisms of available drugs. So this may seem kind of far out there, but there are actually people who are doing this right now. And they're anecdotally, in Vancouver, for example, Mark O'Mara's group has talked about this at some meetings. This actually has borne some fruit in the cases of some specific patients where they did whole genome sequencing. They identified therapies they would not have thought of otherwise, and they applied them, and they worked. So another area I think there may be some potential, at least is interesting to think about, is prenatal screening for trisomies. So there's a paper from Stephen Quake's group a few weeks ago, or in PNAS, basically showing that you could shotgun sequence DNA from cell-free plasma obtained from pregnant women, and because so a substantial fraction of circulating DNA in cell-free plasma is from the fetus, you can actually see an overrepresentation of tags derived from chromosome 21, right? So by sequencing essentially millions of tags, a very deep sequencing. So here on the y-axis, we have the ratio, the relative ratio of tags derived from chromosome 21. And in mothers whose children have trisomy 21, you actually see this ratio is elevated. So it's a very subtle elevation, right? So just a 10% elevation. And from that, you can back calculate the fraction of DNA in the maternal serum that's fetally derived. But it just gets you thinking about some possibilities here. So microbiology is another area. I think there's potentially a lot of possibilities. So there have been many people doing this, but here's a nice example of sequencing of clinical isolates. So basically whole genome sequencing of clinical isolates. And you can construct a phylogenetic tree. You can sort of map when resistance markers entered the genome based on this phylogeny. And you can imagine that could be, it's interesting from a research point of view, thinking about the evolution of salmonella here, but you can imagine how that might be clinically useful as well, or epidemiologically useful. So right now, if I had to ballpark the cost for resequencing a single microbial genome, it's probably about $500. So that's maybe too high to think about doing this at scale. But things are evolving rapidly enough that one could think about, or start to think about, when does the cost become low enough that we just sequence the genome of every bacterial culture that comes through the lab, right? And why bother? So one, essentially, this would enable comprehensive molecular epidemiology. So even if it wasn't relevant for a specific patient, you would have a sense of what was going on across the hospital or the community. And potentially, as we learn more about the determinants of antibiotic resistance, this would be a way of doing that as well, right? So another area that I think has a lot of potential, maybe over a slightly longer time frame, but lots of potential nonetheless, is profiling of the immune repertoire. So a very nice paper, again, from Stephen Quake's group, and this was focused on a model organism, Zebrafish, which has a much simpler immune system than a human. But essentially what they're able to do is to deeply sequence the VDJ repertoire of these individual zebrafish and get essentially a comprehensive molecular profile. Other groups have done this in humans and with interesting results. And you can imagine that this would be a very sensitive way to detect and then monitor clonal expansions broadly across immunoproliferative disorders. And so here we're seeing the axes here correspond to VD and J. And this is sort of this is a zebrafish, but you can imagine an equivalent graph for humans, for a human that would be perturbed in the case of a clonal expansion. And then could be followed up on over time. So the last area I'm going to talk about here is medical genetics. And here I'm going to transition to actually telling you a little about some of the research that we're doing, specifically on exome sequencing. So the exome, or the term exome, corresponds to the protein coding genome. So here we're just talking about the approximately 1% of the human genome that codes for proteins, unhelpfully split across over 100,000 exons. We include in our definition canonical splice sites and microRNAs, but exclude the untranslated regions of transcripts. And the basic idea here is that whole genome sequences are great, but they're very difficult to interpret, especially in non-coding regions. Whereas the protein coding region is eminently more interpretable, is enriched for variants that have large effect sizes, are easier to follow up on. And because we're only focusing on 1%, this costs much less than conventional or next generation whole genome sequencing. So there are some costs incurred with trying to target just the exome. So it doesn't cost 1% of sequencing a whole genome, but it does cost about 5%, so about 20-fold less than a whole genome. So the technical challenge here is that there's no efficient way to, or until recently, we lack efficient ways to selectively isolate the fraction of the human genome that is protein coding. So with conventional Sanger sequencing, your Sanger sequencing read was 1 kilobase, your PCR amplicon is 1 kilobase, and that was a perfect match. But with next-gen sequencing, we're sequencing 100 million or a billion bases at a time. How do you specify what 100 million or what billion bases you want to sequence in a genome? So there are lots of approaches to do this, some of which we've contributed to. The one I'm going to focus on, because the data I'm going to show you was obtained by this method, is relatively simple, array hybridization. So the basic idea here is to use a microarray in a preparative fashion rather than in a interrogative fashion. So we make a shotgun genomic library. So we chop up DNA, put on adapters, amplify that up, and we order a programmable microarray from Agilent or Nimblegen. And the features on that microarray correspond to human exons. And we simply hybridize the shotgun library to the array, and that enriches for sequences that are derived from the exons. And then we elute the specifically bound material off the array and sequence that on a massively parallel sequencer. So if there are any people in the audience who want some more specifics, what we're actually targeting here is approximately 28 megabases that corresponds to a relatively conservative definition of the exome. We use either two 244K or one million feature array from Agilent. The input requirements are about 5 micrograms. All of our sequencing is being done on Illuminas with 76 base pair reads. We typically obtain about 6 gigabases of sequence per individual. Half of these reads map to exons, half of them don't, but that's still a 50-fold enrichment because at random you'd expect only 1% to map to exons. And they're relatively uniform in terms of their distribution across different exons. So here's a screenshot of a particular region of the genome. So here at the bottom we have, in the blue boxes, correspond to individual exons. And as you can see, the peak heights correspond to where the sequencing reads map. And so we get these sharp spikes over the exon. So our reads are selectively being enriched for exonic sequences. And we do have this low background of reads that map to other locations. So again, I showed this slide earlier. The goal is to discover bona fide variants. And in particular, in the context of what I'll be telling you about, we're looking for variants that are relatively rare in the human population. But in a given individual, for example, would be 50-50 for a heterozygous variant. So we need multiple reads to cover a given variant in order to be able to call it. So we require at least 8 reads to cover a given position. Right now, with the data I'll show you, for each exome, we're covering about 96% of protein coding sequences with sufficient depth to be able to make a variant call. And in total, we've generated several hundred megabases of coding sequences across multiple individuals. So the way that we quality control is to compare these data to data obtained by genotyping on a Illumina 1 million featured genotyping array, and we get very good concordance. So we know that our calls are quite accurate. So we published this pretty recently in Nature, sequencing exomes of 12 individuals that I'm showing here, and we can estimate it. We can essentially calculate the total number of non-synonymous variants across the genome, and you see it's very consistent by population. So these are 6 Europeans, 2 Asians, and 4 Yorubans, or African descent individuals, and it basically corresponds with what we expect based on the expectations for these populations. So the data I'm going to show you next relates to a Mendelian disorder. So why study Mendelian disorders? So as I alluded to earlier, there's a disconnect between personal genomics and actually translating this to something that's disease relevant. So we thought a nice approach to start with to show how this could be applied in a meaningful way would be to focus on a Mendelian disorder. This is the simplest case you can imagine for genotype-phenotype, monogenic traits passing through a family. And this is the foundation of human genetics, essentially. 3,000 approximately Mendelian diseases have been solved using classical linkage analysis or a candidate gene approach, but thousands of diseases remain unsolved, largely because we lack the pedigrees, sufficiently complex pedigrees to be able to map the traits with sufficient resolution. So we decided to take a different approach to this and basically ask, can exome sequencing be applied to identify the cause of a Mendelian disorder? So this is a collaboration with a number of individuals here at the U, including Mike Bamshad and Debbie Nickerson. And we started with actually something where we knew the right answer. So this is Freeman-Sheldon syndrome. It's a congenital contracture syndrome. It's known to be autosomal dominant, and Mike Bamshad's group showed in 2006 that mutations in MYH3 underlie this disease. And this, again, was a case where they couldn't have solved this through a classical linkage analysis because they lacked the families, but rather they solved it through a candidate gene approach. They sequenced a few genes they had a good guess about, and they got it right. So knowing the answer, we went into this with the following approach. We sequenced the exomes of 4 individuals affected with FSS, or Freeman-Sheldon syndrome, and 8 individuals who were unaffected, and these are individuals from the International HapMap project. We then applied a filters where we removed common variants under the presumption that variants that are common in the human population are unlikely to underlie a Mendelian disease. And finally, we looked for genes where all individuals affected by FSS contained uncommon variants, right? Under the assumption that this was a monogenic disease with 100% penetrance. So I'll show you how the data looks here. So basically, in each column here, we're looking at the addition of an additional individual with FSS to the data set. And we're basically asking how many genes are there in the genome with, that meet a certain set of criteria. So if we take all comers, if we don't try to discard common variants, even with 4 individuals, there's still a remarkably large number of individuals where all 4 individuals have some sort of variant that affects coding sequence. Now, if we use public SNP databases as the filter to remove common variants, this essentially is pretty powerful. So with just 4 individuals, we can get down to 53 candidate genes for the disease, where all 4 individuals have a variant that is non-synonymous and not represented in dbSNP. So we also sequenced these 8 individuals from the HapMap project, or sequenced their exomes, and so that essentially provides a filter as well. If we put aside dbSNP and just use this as a filter, remarkably, just these 8 individuals are actually more powerful than dbSNP as a filter. So we're down to 21 candidate genes with this filter alone. If we put these 2 filters together, there's only a single gene, which we know to be the cause of Freeman-Sheldon syndrome, that contains rare variants or variants not represented in these two databases in all 4 individuals, right? So the answer is essentially yes, and we've subsequently gone on to try this on. We've only tried one Mendelian disorder of unknown origin. We're 1 for 1. So, and the plan now is to extend this to more diseases. So how is this relevant in the clinic, or is this relevant in the clinic? So you can imagine that exome sequencing would essentially constitute a single test for known genetic disorders. So even though you suspected X, you might as well just sequence the whole exome because it's cost effective to do so, and you might be wrong. But perhaps more importantly, kids with potential genetic disorders but no definitive diagnosis, this might be the way to go. So both as a diagnostic and also as a research learning tool, we think there's some potential here. So some challenges, I think, include the incompleteness of the data. So we were very proud of our 96%. In talking to people in medical genetics who are focused on resequencing of particular genes, they often want 99.9%. And so that's using next-gen technologies, that can be challenging. And I think that's something that needs to be thought about. Also, by focusing on the exome, we're by definition excluding non-coding variants. And so, but on the other hand, those are difficult to interpret, and so it's just an issue to think about. More importantly, or quite importantly, I think variants of unknown significance are going to be popping up all over the place, and I think this is going to be a continuing challenge that needs to be thought about. And one thing that we, in the data that we've collected so far, we haven't gone and checked if these individuals had disease X or Y or Z, because that would essentially result in incidental findings, some of which can create some difficulties in terms of figuring out what to do, whether to reapproach the patients when they're actionable, whether not to. And these are just issues that I think are going to be coming up increasingly as we think about whole genome sequencing or whole exome sequencing at broader scales. So just to kind of wrap up with a few slides here. So massively parallel sequencing of essentially constitutes having these dense arrays of sequencing features and obtaining data by some biochemical reaction. So for example, polymerase extension with fluorescent nucleotides, followed by serial collection of images, and then analysis of stacks of images over sets of cycles to extract sequence data. It has a very low cost per base, but lower accuracy and shorter reads than conventional Sanger sequencing. And I think across these broad areas that I talked about, there's a lot of potential for clinical applications. Not necessarily in this idea of personal genomics, but I think in these other areas that where the results might be achieved in a much more tangible and short-term way. And so I thinking broadly about the challenges that are going to be faced in actually trying to realize these in a clinical lab. Implementation is obviously going to be a very large challenge. These are technically these are still technically problematic platforms. They're changing very quickly and becoming more robust, but there's still a lot of issues, and there's quite a bit of computation involved. It's certainly a solvable problem, but I think that the critical mass of people who know how to really use these things is still quite small. It's growing rapidly, but that's still something we need to make some progress on. I think maybe in 2005 or 2006, there were probably less than 10 of these instruments out there in the community. Now there are well over 1,000 just in those 3 years, and so this is a field that's taking off pretty rapidly. I think we need more clear demonstrations of clinical utility. So in order for this to really be worthwhile, it needs to add value and also be cost effective. And then I think, I talked about this a little earlier in the context of medical genetics, but I think it's going to have relevance for some of the other applications as well, is incidental findings are going to crop up frequently, and thinking about those in advance and whether to do consenting in an appropriate way and that sort of thing. And then finally, findings of uncertain significance are going to be a large issue that, again, I think we need to think about as a community and figure out what the appropriate guidelines are for that. So end by thanking a few people, or quite a few people. So the exome work that I showed, again, this is team science, and some of the key people involved are Sarah Eng and Emily Turner in my lab, as well as Debbie Nickerson and her group, and Mike Bamshad and his group, all here at the U. And lots of other people are involved in this work as well. So thank you very much, and I'd be happy to take any questions. So,
the work that you did on the Mendelian trait, tracing that one down, I guess it was a myosin gene, I don't know about getting that, obviously it's a little premature as a clinical test, but in terms of a research, let's say someone had a clinic where they followed patients where it was unknown, and they decided in my research lab, I do want to apply this technology. Is there any sense that you could have obviously at the first one, it was not representative of how it might work in the future, but how long does it take and how much does it cost to actually do all those sequences that would need to be done, analyze all the data and all that?
So it's evolving very quickly. The work that I talked about was largely done by a graduate student over the course of a few months, and one technician running an instrument. We're in the process with Debbie and others of scaling this and trying to optimize some aspects of it. So I think it's fair to say $1,000 to a few $1,000 per individual. I think to really get a research finding of meaning, you need multiple individuals. And it could be done in the order of a few weeks from the time of actually getting the DNA and having it in the queue. So it can be pretty quick, I think. And I think once a pipeline exists, it's very easy to turn the crank, right? The key, I think, the more challenging part, I think, for success there is to identifying patients and phenotypes that are amenable to this approach, right? So we're being very careful about those selections, and I think in some ways that's going to be a large determiner of success or failure.
Yeah. So just for my own personal information, so at the very beginning you're talking about your sequencing, your sort of this array of sequences and adding a base at a time. How is it that you get an individual sequence on an individual spot on the array? How do you get a homogeneous, because I'm assuming this is from somebody's genome, right?
So you're asking, so each spot on the array is thousands of copies of a single fragment of DNA, right? That's been amplified in some way so that copies stay locally clustered. You imagine having a common sequence, unknown sequence. You have a primer, right? You have a polymerase, 4 fluorescent nucleotides. The nucleotides are reversible terminators. So only a single base will add, right? And then it'll stop.
But that part I get, but just in terms of how you have a uniform sequence on each of your spots.
A uniform. How do you get 1,000 copies at each spot? So there are a number of ways to do this. So the way I'll tell you about how it's done in the Illumina platform. So imagine doing PCR where both PCR primers are actually physically tethered to the surface of the array, right? So a single molecule with adapters floats down, right? Some shotgun sequence, and a PCR reaction initiates. So it extends and then, denature. The next copy is going to bend down. You'll get a local copy, right? And so all the copies stay locally clustered to that single position. Does that make sense? So that's essentially it. There are other ways to accomplish the same thing. Helicose is another company that doesn't even bother to amplify. They just do single molecule sequencing.
So the other question I have is relating to the myosin heavy chain, your patients. Did they all have the same mutation, or do they all have individual mutations just of the same gene?
They had individual. I think some of them were the same, but we allowed for different mutations in the same gene. So some were the same, some were different.
Yeah, I'm wondering what's the status of the high-throughput sequencing platforms in general for quantitative analysis, so to become competitive with arrays for gene expression or for analyzing mixed populations of sequences. So
putting aside cost, from a quantitative point of view, I think the results I've seen are excellent. You have extremely high technical reproducibility, far better than microarrays and reproducibility between labs, which has been an issue with microarrays. And I don't know if I've seen a really clear demonstration of cross-platform reproducibility, but people have shown in spike-in experiments and that sort of thing, you really do get very precise quantitation over a broad dynamic range. And you can also look at low abundance things in a way that microarrays are more challenging, or it's more challenging with microarrays. So I do think there's a lot of potential there. I think the more challenging thing to figure out is microarray costs have come down quite a bit, right? So how low, at what point does it make sense to switch over?
Yeah, John.
Yeah, I particularly liked your last application for tracking down the causes of these Mendelian diseases where the gene isn't known. But once you've done that, can you just make some comments about the state of the art for these computational or other approaches to figuring out? I mean, let's just say you think you have a person who has this disease, you find a variant, and then saying, okay, is that variant pathogenic or not? Because that's often the stumbling block of, as you pointed out, with these variants of uncertain significance.
Yeah. This is a big. So the variants of uncertain significance. The best we can do right now is to apply these tools like polyphen and SIFT and other tools that try to predict the impact of these individual variants. I think it's a real issue, and I don't. The best solution that I think will emerge is going to come from more data, and that data won't come from this study in particular, but rather data from projects like the 1,000 Genomes Project. So you can actually, you can actually say if a mutation is rare, there's a reasonably good chance that it actually is. So a very large fraction of protein coding changes are deleterious, right, at baseline, right, and in some ways, things like the 1,000 Genomes Project, if you show a mutation is quite rare, you have some a priori suspicion of that particular gene. I think putting these things together in some sort of consistent model, we can get a lot more traction than we have already. And the other piece of it, I think, that hasn't been applied as powerfully as it could be is conservation, and nucleotide level conservation across mammals. And this data is relatively new, where 30 or so mammals have been sequenced whole genome, ends up being a very good measurement of fitness effects, right? And so I think that's another. So I think there's definitely room for improvement. I think that what we need is some sort of standard approach, kind of, and a way of dealing with, if I can attach a probability to a mutation, then can that be dealt with clinically, or can that be sort of brought back into how one deals with a patient or talks to a patient? Because I think that's what it's going to be. It's always going to be ambiguous at some level, right? It's just a matter of assigning a probability.
I wonder if you could comment briefly on some of the potential advantages of the third generation proposed sequencing methods that aren't commercialized yet. Are we getting down to, as you said, single molecule sequencing and just more parallelism, or is there something else that they're...
So the question was about the third gen technologies and whether they're going to have an impact. I think the second generation technologies are here to stay for a while. I don't think anything's going to displace these things anytime soon in a very large way, the way that Sanger is being displaced right now. The Pacific Biosciences technology, for example. So I won't go into the details of how this works, but imagine you could get a 50 kilobase read that was terribly inaccurate, right? So on one hand, it's a 50 kilobase read, which is unprecedented. On the other hand, it's terribly inaccurate. But that still has a lot of uses. So people like Evan Eichler, who are studying, complex structural variation, would love that technology, right? For assembling there's a lot of applications where I think it could be really powerful and fill a niche that these technologies can't get at. So I think in the short term, that's going to be more likely where those technologies are initially applied. Full-length mRNA sequencing, things like that.
I'm wondering how this technology will work in the sort of the needle in the haystack problem, and I'm thinking about the issues of a few organisms that are pathologic amongst a whole bunch that are not, or the problem of colon cancer screening, where the sequence of interest is mixed with tons of normal human and bacterial microbes in the middle of a bunch of stool. Will this solve some of that problem of finding the needle in the haystack?
I think it will go along. I think it will. I think it has the potential to solve a lot of those issues. I think the problem of seeing if organism X is present in the complex mixture in some ways is much easier, because you only need to see a few tags to have a fairly reasonable confidence in quantitation of how often it's present, is easier than finding, say, like the data I showed people identifying 2% mutations in EGFR kinase in a background population of 98% normal, right? But that took 50,000 reads to get that 2%, right? So I think for finding mutations at extremely low frequencies, I think is going to be extremely challenging. But I think to the general question of whether first you can detect pathogenic organisms in complex sequence mixtures, I think absolutely there's a lot of possibility there, and also you're opening yourself up to finding other things, right, that you might not have expected, in a way that you wouldn't be doing if you were doing a directed PCR assay or something like that. And then the tumor thing, I think also, no question, I think there are going to be challenges like, trying to get below 1%, I think is going to be extremely challenging. But in general, I think there are definitely some possibilities there.
So I have one question about the granularity problem.
Uh-huh.
Are there any, is that something that you think is solvable, or do you think that the answer is just going to be kind of trying to make use of more information, or can we really break it down?
So the granularity problem, which is that you essentially your minimal unit of sequencing is several hundred million or a billion bases, let's say, and if you want much less than that, for example, per patient. So there are solutions out there that don't work very well right now, or don't work, that are difficult or challenging to implement. I think other people are working on things that will be substantially easier. So I think they're surmountable challenges, but in the immediate moment, it's definitely an issue. There are not a lot of people who are, so barcoding, for example, you can imagine putting on a sequence barcode to individual samples and then mixing them into the same lane of sequencing. And that works, but there's not a lot of people who are doing it well still because there are technical issues.
But you're still
Putting that in the clinic would have its own nightmares, I
You're
think.
still batching specimens, right? And you're batching patients, or
Right. Well, so for this, for the exome sequencing, we need 6 billion bases, right? So it's going to, it's larger than our minimal unit, so that's a good thing, right? If we want to sequence a bacterial genome, we sequence it to 1,000x unnecessarily, just because it's easier than actually splitting it up and making the most use of the territory. So that's, it is an issue for sure.
so yeah. So you can reduce the per base cost.
You
but
can, you can reduce the per base cost, but there's a minimum problem size. There's a minimum size of your if you want to sequence, for example, one gene in 10,000 patients, on paper that looks really cheap. But when you try to actually put that into practice in the context of these technologies, it's extraordinarily difficult because you have to mix all these patients into the same reaction volume, and so you need to figure out a way to deconvolute the information.
Any other questions? Well, let's thank Dr. Shendure again.
Thank you.